The plasma membrane is more than a cellular envelope. It is a master regulator, a sentinel, and a conductor of molecular symphonies. Today, we uncover the structure that orchestrates nutrient exchange, communication, defense, and identity. Fasten your seatbelts. This is a journey to the gatekeeper of life itself. Welcome to EasyPhysiology ResearchPro. By the end of this session, you'll grasp the blueprint of the plasma membrane's architecture, decode the roles of lipids, proteins, and carbohydrates, and explore how structure begets function. We'll reveal how this membrane isn't static. It moves, adapts, and signals, always ensuring cellular survival in a chaotic external world. Think of the plasma membrane as a selectively permeable fortress. It defines identity and order in a sea of entropy. It evolved from Gorter and Grendel's early observations in 1925 to Singer and Nicholson's revolutionary fluid mosaic model in 1972. Different cells tailor it uniquely. Myelin sheaths insulate neurons, while microvilli boost absorption in intestines. This is form-meeting function in real-time biology. The plasma membrane isn't a wall, it's a shimmering sea. Lipids and proteins drift laterally, adjusting to stress, growth, and cell signaling. This dynamic model showcases flexibility at its finest. It's a mosaic, yes, but one in perpetual motion. A vibrant canvas where proteins float in a lipid ocean, fine-tuning every moment of cellular existence. Welcome to the core of the membrane's architecture, the lipid bilayer. It's more than just a wall. It's a self-assembling, self-regulating, selectively permeable force field that protects the cell and enables life's choreography. Let's break it down. At the molecular level, the hero of this story is the phospholipid. Each one is an amphipathic marvel, with a hydrophilic head that loves water and two hydrophobic tails that fear it. When exposed to an aqueous environment, they instinctively arrange themselves into a bilayer, hydrophobic tails tucked inward, hydrophilic heads facing the watery inside and outside. No instructions needed. Nature just assembles. This spontaneous formation is not just elegant, it's thermodynamically favorable. The result? A semi-permeable barrier that protects the cytoplasm, excludes toxins, and maintains ionic gradients. It's the molecular version of crowd control, but phospholipids don't work alone. Enter cholesterol, the membrane's thermostat and stabilizer. Lodged between phospholipids, cholesterol keeps the membrane fluid but not floppy, stable but not stiff. Let's answer a crucial question. What does cholesterol do at different temperatures? Answer. At high temperatures, cholesterol restricts phospholipid movement, reducing fluidity. At low temperatures, it prevents tight packing of fatty acid tails, maintaining fluidity. It's like an internal regulator, adjusting flexibility with environmental changes. Without cholesterol, membranes could either become too leaky or too brittle. Either way, catastrophic. Next, we meet glycolipids, lipid molecules adorned with carbohydrate chains, found exclusively on the outer leaflet of the bilayer. These sugar-tipped lipids play pivotal roles in cell recognition, signaling, and adhesion. Think of them as molecular name tags, guiding immune cells, embryonic development, and even viral docking. Why are glycolipids important? Answer. Because they are essential for cell-cell communication, immune recognition, and the creation of the glycocalyx, a sugar-rich protective coat that guards against mechanical and chemical insult. Now consider this. If we replaced unsaturated fatty acids in the membrane with saturated ones, what would happen to the membrane? Answer. The membrane would become less fluid and more rigid. Our explanation? Saturated fats have straight tails that pack closely, reducing mobility. Unsaturated fats, with their kinked tails, introduce space, enhancing flexibility. So membrane fluidity is, quite literally, a matter of lipid geometry. To summarize this powerhouse slide, phospholipids build the foundation. Cholesterol fine-tunes the fluidity. Glycolipids decorate and define the cell's external identity. Together, they form a living, dynamic barrier, the first line of defense and communication. This bilayer isn't just alive, it's smart. Proteins in the membrane are not just fillers, they're the true workhorses. From integral receptors detecting signals to peripheral scaffolds maintaining structure, they customize each membrane's role. Their asymmetrical orientation ensures precise control, inside and out. This heterogeneity is what makes membranes multifunctional marvels. Now enter the sugar codes, glycoproteins and glycolipids that crown the membrane with the glycocalyx. These sugar chains do more than decorate. They dictate cell recognition, modulate immune interactions, and act as docking stations in signaling cascades. The glycocalyx is your cell's signature, its social ID, its immune passport. This membrane doesn't just allow or block, it discriminates with exquisite precision. Tiny non-polar molecules breeze through. Charged ions? Not so lucky. Selective permeability isn't a flaw, it's an evolved survival trait. It's how the cell maintains homeostasis amidst environmental chaos. Passive transport is diffusion's masterpiece, simple, elegant, energy-free. Oxygen and carbon dioxide drift effortlessly. Glucose uses facilitators. Water obeys osmotic gradients through aquaporins. No energy expended, yet so much achieved. It's kinetic grace in cellular transport. But sometimes, the cell must push against nature's flow. That's where active transport dominates. Using ATP, it pumps ions like sodium and potassium to sculpt electrical gradients. In intestines, it piggybacks glucose using sodium gradients. It's strategic, energy-intensive, and vital. Transport proteins are precision gates. Channels open selectively, carriers reshape to ferry molecules. ATP-powered pumps go against gradients like cellular Olympians. GLUT1 for glucose, sodium-potassium pumps for voltage. Every transporter is a guardian of cellular balance. Receptors are the cell's ears. GPCRs, RTKs, they sense extracellular whispers and shout intracellular instructions. From growth to gene expression, it all starts at the membrane. Ligand binds, conformational changes ripple, and signal cascades roar. Biology, digitized at the membrane interface. Cells don't live in isolation. CAMs build tissue structure. MHCs ensure immunological surveillance. Gap junctions synchronize the heartbeat. Tight junctions keep barriers leak-proof. Desmosomes hold it all together. This isn't just communication, it's molecular diplomacy and defense. Fluidity is life. Saturated fats stiffen the membrane, unsaturated fats keep it flowing. Cholesterol fine-tunes this dance across temperatures. Cold? Add kinks. Hot? Add control. Organisms tune membrane fluidity like symphonies respond to tempo. The two faces of the membrane are not mirror images. The outer leaflet signals the world, the inner leaflet handles logistics. Flipases, flopases, scramblases orchestrate this choreography. Asymmetry isn't just elegance, it's essential. Apoptosis begins with asymmetry gone wrong. Adaptability breeds specialization. Microvilli multiply surface area. Myelin accelerates electrical signals. Lipid rafts become command centers for receptors. The basic membrane evolves into a multifunctional specialist. It's tailored architecture for tailored function. Diseases begin at the membrane. CFTR mutations cause cystic fibrosis. Lupus targets phospholipids. HIV exploits CD4 and CCR5. The membrane isn't just biology, it's pathology, diagnosis, and therapy. Understanding it means controlling disease at the cellular frontline. Science unravels the membrane using cutting-edge tools. Freeze-fracture microscopy reveals its topology. FRAP quantifies diffusion in real time. Patch clamp dissects ion channels one by one. These aren't just methods, they are microscopes into membrane magic. The plasma membrane is nature's paradox, structured yet fluid, selective yet communicative, barrier yet gateway. It adapts, signals, transports, and protects. A true biological marvel balancing identity, exchange, and survival. In understanding the membrane, we understand the cell. Now that we've decoded the secrets of the plasma membrane, let's bring it all together with a few high-impact conceptual questions, designed not just to test your memory, but to ignite your critical thinking. Let's begin with a multiple-choice warm-up. Question 1. What would happen to membrane fluidity if the proportion of saturated fatty acids increased? Answer. Membrane fluidity would decrease. Our explanation? Saturated fatty acids have straight hydrocarbon chains that pack tightly together, making the membrane more rigid and less dynamic. In contrast, unsaturated fatty acids contain one or more double bonds that introduce kinks, preventing tight packing and enhancing fluidity. So, if a membrane loads up on saturated fats, it becomes stiff, less adaptive, less functional. Imagine a tightly packed crowd at a concert versus one with space to groove. That space is crucial for membrane flexibility. Question 2. Which transport mechanism would be most affected by ATP depletion? Answer. Primary active transport would be most affected. Our explanation? Primary active transport mechanisms, such as the sodium-potassium ATPase, directly consume ATP to move ions against their concentration gradients. Without ATP, these pumps grind to a halt. That means neurons can't generate action potentials, kidneys can't reabsorb sodium, and your entire electrochemical system loses balance. Passive mechanisms like diffusion or facilitated transport don't need ATP. They'll continue, but only if concentration gradients exist. Question 3. What role do glycoproteins play in viral infection? Answer. Glycoproteins serve as receptors or docking stations for viruses. Our explanation? Many viruses, including HIV and influenza, exploit membrane glycoproteins to gain entry into host cells. For instance, HIV binds to the CD4 glycoprotein and a chemokine coreceptor like CCR5. Without these specific glycoproteins, the virus cannot attach or enter. In some people, mutations in CCR5, like the delta 32 deletion, confer resistance to HIV infection. In essence, your membrane's sugar-crowned proteins determine whether you're an open door or a locked vault. Now, let's raise the stakes with case-based applications. Case 1. A patient has abnormal red blood cells with increased sodium permeability. What membrane component might be affected? Answer. Integral membrane proteins, especially ion channels or sodium pumps, might be dysfunctional. Our explanation? The increased permeability suggests either leakiness through defective channel proteins or failed active transport due to sodium-potassium pump impairment. Structural proteins anchoring the cytoskeleton, like spectrin, may also be compromised in conditions like hereditary spherocytosis, altering membrane integrity and ion balance. Case 2. Arctic fish survive in near-freezing waters. Predict adaptations in their membrane composition compared to tropical fish? Answer? Their membranes likely contain more unsaturated fatty acids. Our explanation? In cold environments, membranes risk becoming too rigid. Unsaturated fatty acids help maintain fluidity by preventing tight phospholipid packing. It's a brilliant evolutionary trick. Swap out straight-chained lipids for kinked ones and keep the membrane fluid even when temperatures drop near freezing. Tropical fish? They're tuned the other way, with more saturated fats for membrane stability under heat. Homework prompt. Explore a membrane transport disorder. One fascinating example? Cystic fibrosis, where mutations in the CFTR chloride channel disrupt ion transport and water movement, leading to thick mucus buildup in lungs and intestines. Understand the gene, the protein, the clinical consequences, and how this single transporter's failure wreaks systemic havoc. You'll realize, mastering the plasma membrane isn't just academic, it's clinically transformative. In conclusion, this membrane isn't just a cellular structure. It's a battlefield, a blueprint, a laboratory. Every protein and lipid tells a story. So, keep asking. How does structure determine function? How does function determine fate? Stay curious, stay inspired, And remember, the membrane is life's first interface with the world. Treat it with the scientific respect it deserves, and subscribe to Easy Physiology and ResearchPro for more physiological concepts.